All right, good morning. I want to record this weekend's videos early because we have a test on Monday. I'm going to go ahead and talk about the case study. This was at the very end of class on yesterday, which was Wednesday. I can't even remember what day it is. But we had a case study that we talked about, and I wanted to mention this to you. I wanted to go through this. The nice thing about doing this by a video is that you can do it at your own pace. I'm going to ask you some questions. And if you treat it like a test question, you sit there and you think about the data and interpret it, then I'll go on and I'll mention the answer. So use it as you will, but I think that this could work out nicely for you. So let's talk about the case of Helen Burns. This is a 6-month-old child, and she has a immunodeficiency. She was basically about 6 months of age. She developed pneumonia in both lungs, severe cough, and fever. This is unusual. They cultured the blood and sputum for the presence of Pneumocystis carinii. And so it's an anti-pneumocystis drug, and that specific antibiotic, as usual, worked well to remove the infection from her body. But most 6-month-olds do not get Pneumocystis carinii. Usually the immune system can fight it off, so it's an opportunistic pathogen. And it was suspected that Helen had something wrong with her immune system, something missing. So, the peripheral blood mononuclear cells, these will include the lymphocytes, right? They were collected, and this is basically you're looking at her adaptive immunity. And you test those in vitro. So they tested how her T cells grew, and they grew naturally. By the way, the traditional way to do this is to incorporate tritium-labeled thymidine. When they're growing, they need more DNA, and thymidine is the building block of DNA. And so they incorporated the thymidine at a normal rate, and they stimulated it with a PAMP-like molecule, so like a pattern associated with molecular infections, and it actually will cause T cells to grow when you give it. So I just wanted to tell you that these are useful in the lab. This is how they actually do it in clinical chemistry. So she had been given her DPT vaccine, but her T cells, and so they tested her T cells against tetanus toxin. It did not respond to tetanus toxin. That means there's something wrong with the T cells. And by the way, her T cells did respond to allogeneic B cells, so it was not some general problem with the T cells. It was specifically to the way that they were responding to the tetanus toxin. So the question is, are these cytotoxic or helper T cells that are having the problem? And of course, you're going to measure immunoglobulin levels to see what antibodies are like for her. We're going to talk about the differences between the classes coming up. But for right now, they just had these 3 classes, and you can look at the range they reported and the normal range for each of it. IgG 96, which is the normal range is 600 to 1400. IgA was 6, with normal range was 60 to 380. IgM was 30, normal range was 40 to 345. So take a second and look at those and say, okay, is this in the normal range, or is this a particular direction from the normal range? If you receive these reports back from a patient, what do you say about their antibody levels? And so I think that you can see that they're all lower than they should be. Okay, so then you count the white blood cells. The white blood cell count was 20,000 as opposed to 4,000 to 7,000 is normal. So make a note of of how that fits into the normal range. And 10% of those were lymphocytes. So you look at the lymphocytes. So there were definitely outside normal levels when you look at that. So about 3,000 is normal, so you can do the quick calculation of about where are you for white blood cells in general, and where are you for lymphocytes. The lymphocytes were tested with antibodies, and so you take, by the way, the antibodies, the way you tell, traditional again, the way that you tell which lymphocyte it is, you can't tell by looking at it under the microscope, right? So you give it an antibody to a particular marker. And there are certain molecules that are expressed on the surface of B cells, different kinds of T cells. You define the cell by these markers. They see that this protein is always on B cells, And so they see CD20 is a molecule that's always on B cells, and so you use an anti-CD20 antibody to say how many of our cells have this marker on them. And so of the lymphocytes, they tested those. So, you have the 2,000 cells per microliter that were going to be, that were lymphocytes. 7% of those had the CD20 marker that indicated that they were B cells. The normal range is 10 to 12%. 57% of those had CD3, and therefore they were T cells, not B cells. And out of those, you can take that and you can see 34% has CD8, which was 388 cells per microliter, which means it was normal. That's within the normal range, but only 20% had CD4. Those are the helper T cells, and those were 228 cells per microliter. Now, helper Ts should usually be twice as many helper Ts as there are killer Ts. And it turns out that that does not appear to be the case. So write this all down. What's normal? What is abnormal? Make a list, and then say, as a doctor, what are you going to say for what these things, what's normal and abnormal about the patient? What is she missing? And overall, I'll give you the answer now. Antibodies and B cells and helper T cells are missing. I don't know exactly why the white blood cell count is so high. I think that there might be white blood cells like granulocytes maybe that are amplified, but we're not focusing on those. We're focusing on what's missing. And so focus on the important point. The key point is that these three kinds of things are missing. And because we talked about this in class, the connective point between all three of these is they really depend on MHC class 2. You have to have antigen presentation on MHC class 2 to get antibodies from plasma cells, to turn B cells into plasma cells, and the helper T cells are the two-factor identification that's required for that. So the doctor recommended a bone marrow transplant. Clearly you have some kind of deficiency in what's making your lymphocytes, and so you transplant bone marrow that is sufficient to make the lymphocytes. There's some kind of gene probably that's messed up. And if you give it, you can transplant bone marrow if everything matches up on the immunological markers side of things so that the body doesn't reject it. So transplants will require the same MHC class 2 or HLA gene region. They need the same antigen presentation because the whole immune system is based on T cells recognizing the MHC class 2s that you were born with. So the T cells that you have will need to see the MHC class 2s. The good news is there's enough diversity that you can usually find someone who matches you close enough, but you have to go looking because there's so much diversity. Okay, remember that there's three different MHC class 2s, DQ, DR, and DP, and you get 2 copies of each, one from your mother, one from your father. So you can have a maximum of 6 different ones. So they have a way of typing whether you have, like, which type of DR do you have, which type of DP do you have. And they did the typical test, and they were looking for which kind of DR do you have, and the answer came up 0. It's not you have a particular type of DR, it's that you don't have any DR at all. And so the more sensitive, so they used a more sensitive test, that's more expensive and more time consuming, but also more sensitive, called fluorescent activated cell sorting, which is FACS. And what this is it just quantifies how much of each marker is on the cell. I like this because actually, usually our FACS output now is multi-dimensional. But this is an old example, and so it has old data. But let's start with the old data. It's good. This is one-dimensional FACS data. And what it does is it just has a count. The y-axis up and down is how many cells are fluorescing to have that marker on them. And then the number of cells is on the y-axis. If you have more cells that have that marker, so you're counting the cells, and so the farther out you get, the more cells you have, and the farther up you get, the more marker you have. Okay, so you have the results down here for HLA-DQ and HLA-DR. Normal cells are on the right. And so you'll see a lot of transformed B cells. So you see down at the bottom, it's a log scale, but you can see you have about 1,000 transformed B cells that are expressing good amounts of HLA-DQ. We're just looking and seeing that it's there, you know. So this is more about the x-axis than it is about the y-axis. All the y-axes are about the same. And so if you look down on from that HLA-DQ, you get about 1,000 on the x-axis for that in normal cells. If you look at the bottom right, you'll have HLA-DR is about the same, 1,000 for normal cells. But if you have a class 2 deficiency in a particular gene, then your cells will be more shifted to the left. You'll see much fewer cells. Not 0, but basically 10, or maybe up to 100 rather than 1,000. Okay, so you have the cells with the cells on the left. You can see that those are definitely shifted to the left. And so you have much fewer HLA-DQ. That's an HLA-DQ deficient patient. The HLA-DR deficient patient, which would be like what Helen's blood would show, is shown on the bottom left. Don't overcomplicate these graphs. Really, all they're saying is that you have basically the number of cells on the x-axis. The farther to the right, the more cells have that thing. The immunodeficient patients hardly have anything. Facts showed that she didn't have the HLA-DR, and if these came from her, then they would be like she would not have HLA-DQ either. It'd be kind of cool if they showed an HLA-DP just to show if she had that, because if she had that, the HLA-DP would have the red peak around 1,000, 10 to the third. It would look like the panels on the right. By the way, I'm also going to include figure captions. If you need to, pause and read those captions. It's actually good practice for reading technical language. But the conclusion is that, okay, it looks like a bone marrow transplant will work, and we just need to make sure that the bone marrow person, we don't need to worry about matching up the HLA-DR. We just need to make sure that the person is as close as possible to having the same types. And there's a possibility of it rejecting by reacting to a new HLA-DR, but hopefully the immune system will be able to adapt rather than expecting. It's not expecting an HLA-DR at all, so I think that in this case a bone marrow transplant is fine. And so they tested her mother, but they tested her brother first. Her brother did not have the same HLA typing, did not have the same DR, DP, and DQ molecules. So her mother was a closer match, had a better match to that, and that makes sense because you're probably going to have half the genes the same if you're going from parent to child, at least. And so I just want to show you, you give her a specific cytotoxic drug to sort of bring down the whole immune system. So busulfan is given to depress bone marrow function. And then there is cyclophosphamide, which is really nasty stuff. It will ablate her bone marrow, basically getting rid of it. Then her mother had to have her bone marrow depleted of T cells so that her mother's T cells wouldn't come and react to Helen's body. The graft was successful and the immune function was restored. But because you have to destroy the marrow that's there before you add transplant in new marrow, bone marrow transplants are a really risky proposition. But in the case of this, it would prevent Helen from continuing to get opportunistic infections every single time. Okay, so that's a bit of detail, but I want you to be able to solve cases like this. You know, I will give you like the pharmaceutical details. You don't need to remember what the name of the drug is, but the purpose of the drugs and the purpose of the different tests and things like that, being able to give you a normal range and for you to say, hey, this is normal or this is abnormal, because that's exactly what you will do if you ever go into the medical field. All right, so that's our case study. That will finish up our lecture material, and now I just have a journal club to give you.